This side-by-side time-lapse shows the stark difference in cellular lifespan between our two primary nucleic acids. On the left, a DNA double helix remains intact, retaining its sequence through multiple cellular divisions. On the right, a strand of messenger RNA degrades and fragments into single nucleotides shortly after it's synthesized. Biology demands this discrepancy. DNA sits protected in the nucleus, storing the genetic blueprints required for an organism's entire lifespan. mRNA carries a temporary, single-use instruction to the ribosome and must be quickly destroyed to prevent runaway protein production. Yet, if we examine their atomic composition, DNA and RNA are almost indistinguishable. They are both linear chains of 5-membered sugar rings linked by phosphate groups, carrying the same basic alphabet of nitrogenous bases. This divergence in biological function, permanent archive versus disposable message, originates from a specific physical organic chemistry constraint involving a single oxygen atom. To locate that atom, we have to look at the continuous chains holding these molecules together, their sugar-phosphate backbones. Both polymers use a repeating 5-membered ring sugar as the central structural link in this chain. Here is the structural difference. The sugar on the left, D-ribose, is found in RNA. It has a hydroxyl group on its second carbon, known as the 2' position. The sugar on the right, 2'-deoxy-D-ribose, is found in DNA, and it lacks this oxygen atom. The presence or absence of that specific hydroxyl group determines the fate of the molecule. To see exactly why this one oxygen atom leads to destruction, we must construct the nucleic acid polymer from first principles and observe how the individual pieces interact. We start with the information carriers, the nitrogen-containing bases. These come in 2 structural variations. Purines feature a 2-ring structure, while pyrimidines consist of a single ring. This image maps how these bases attach to the sugar ring. The connection forms at the anomeric carbon, the 1' carbon of the sugar. It links directly to a nitrogen atom on the base, nitrogen 9 for a purine or nitrogen 1 for a pyrimidine. This connection relies on strict stereochemistry. It forms a beta-glycosidic linkage, where the nitrogenous base and the substituent on the 4' carbon are positioned on the same side of the furanose sugar ring. A single base bonded to a 5-membered sugar ring in this specific configuration is called a nucleoside. Looking at the structures here, we introduce the final piece required for polymerization. A triphosphate group is added via an ester linkage to the 5' carbon of the sugar. The model on the far right shows adenosine 5' triphosphate, or ATP. Once the base, sugar, and triphosphate group are fully assembled, the unit is a nucleotide. The addition of those 3 phosphate groups creates a dense cluster of negative charge. These triphosphorylated monomers now hold the thermodynamic potential energy required to drive the synthesis of a long polymer chain. 2 outer phosphate groups are eliminated as pyrophosphate, leaving a stable phosphodiester bond that securely links the sugars. In a cellular environment, the expelled pyrophosphate is immediately hydrolyzed by water into 2 separate phosphate ions. The destruction of this leaving group makes the entire addition process thermodynamically irreversible. Because the mechanism relies on the existing 3' hydroxyl attacking the 5' triphosphate of the incoming unit, the polymer can physically only grow in one direction, 5' to 3'. This directionality and immediate irreversibility guarantee that the chain continuously synthesizes forward, preventing the genetic code from spontaneously depolymerizing back into solitary monomers. We now return to the initial paradox. If DNA and RNA share this exact same phosphodiester backbone, why does RNA degrade billions of times faster? The answer lies with the RNA ribose ring and the extra oxygen atom of the 2' hydroxyl group, now trapped within the crowded environment of a fully polymerized backbone. In this diagram of the RNA backbone, look closely at the spatial geometry. That 2' hydroxyl group is perfectly positioned to act as an internal nucleophile. The oxygen from the 2' group executes a nucleophilic attack on the adjacent phosphorus atom in the backbone. This attack forces the formation of an unstable intermediate known as a 2', 3' cyclic phosphodiester. To resolve this instability, the adjacent phosphoester bond connecting the next nucleotide snaps. The RNA strand cleanly severs itself. DNA avoids this fate. Because it uses 2' deoxyribose, it lacks the hydroxyl group. Without that internal nucleophile present, the self-cleavage mechanism is chemically impossible. The rapid degradation of RNA results from the internal nucleophilic attack of its 2' hydroxyl group. In DNA, the absence of this group prevents this specific pathway, preserving the backbone for the life of the cell. Chemical stability secures a single strand of DNA, but heredity requires a secondary structure to safely store and replicate that information in 3 dimensions. This image displays the resulting double helix. It consists of 2 anti-parallel strands running in opposite directions, with the robust sugar-phosphate backbones facing outward, leaving the bases protected on the inside. The width of this double-stranded molecule is constant across millions of base pairs. To maintain this uniform geometry without bulging or pinching, a larger 2-ring purine on one strand must always pair with a smaller single-ring pyrimidine on the opposite strand. Beyond physical size constraints, these bases pair through specific hydrogen bonds. Adenine forms exactly 2 hydrogen bonds with thymine, while guanine forms exactly 3 hydrogen bonds with cytosine. This precise geometric matching works because the heterocyclic bases exist in their keto-tautomeric forms rather than their enol forms. The placement of the keto oxygen atoms provides the specific hydrogen bond acceptors necessary to lock the base pairs together. The genetic code's fidelity is guaranteed by exact hydrogen bond matching and strict physical space constraints. Horizontal hydrogen bonds dictate the sequence, but they are not the only thermodynamic forces holding the helix together. The planar aromatic bases stack vertically on top of one another. This dense arrangement creates mutually induced dipoles between adjacent pairs, generating strong cumulative attractive forces known as stacking interactions. Finally, at physiological pH, oxygen atoms along the helix's sugar-phosphate backbone carry a negative charge. This continuous wall forms an electrostatic shield, repelling external nucleophiles like hydroxide ions, preventing them from cleaving the vital bonds. The stability of the DNA genome results from the removal of the 2' nucleophile, base pairing in the keto form, and the exterior phosphate electrostatic shield. RNA sacrifices these defenses to remain a flexible, single-stranded, and reactive molecule, attributes necessary for a temporary cellular messenger, but unsuitable for a long-term archive. The biological preservation of complex life across millions of years is the result of steric constraints and ester stability.